Everyone, today we're going to look at our next topic, which is how we manipulate DNA. So today we're going to look at some of the tools and techniques that allow us to be able to do that. From the study design, this is our key focus for today. We're going to look at how we use enzymes to manipulate DNA, specifically how we synthesize DNA, how we join DNA, and how we cut DNA using some different techniques. Before we get stuck into what those techniques are, it's important for us to know the power that manipulating DNA can give us as scientists. So if we can manipulate DNA, we've got lots of different possibilities. We can remove genes, change genes, or even introduce new genes into the genomes of different individuals. We're also going to look at a new technology that's used to edit the genome, known as CRISPR-Cas9, a little bit later on. When we manipulate DNA, we're able to research genes and find out more about their function. We're able to introduce mutations and see what that does to an organism's genome. And we're able to sort of snip out faulty genes that potentially are not working or causing adverse reactions in the body. We're able to activate or repress genes, edit faulty ones, or add new genes to a genome. So this gives us some really great possibilities in order to be able to treat certain diseases as well. This is an overview of the different techniques that we're going to cover throughout this topic. Today we're going to focus, as I said, on how we cut DNA, how we join DNA, and how we make DNA. Later on, we're going to look at how we can separate and sort out different DNA fragments, how we can find them, and how we can amplify to make more copies of particular DNA fragments. Before we talk about how we make DNA, it's important to know how we actually find that DNA as scientists in the first place. So in order to find or locate a specific sequence of DNA that we want to investigate, we use what's called gene probing. So a probe is a single strand of either DNA or RNA, depending on what nucleic acid that we're looking for, and it's actually got a radioactive or fluorescent label on the nucleotides. So the DNA strand of interest is heated up, first of all, to make it single-stranded, and then the probe will bind to that complementary sequence. So it's complementary to the sequence that we're looking for. So we can see a little diagram just below here, where we've got our DNA probe with its little flag, which is indicating its radioactive or fluorescent label. Okay, we insert that probe into either our test tube or into our an organism, and it will then bind to a complementary strand or a complementary gene of interest that we want to find. So that allows us, in the first instance, to find our piece of DNA. Once we've found our DNA, we've got some different ways that we can then make more copies or synthesize more copies of our DNA. Let's have a look at the tools for that. So once we've got hold of our piece of DNA, we've used our probe to locate it. There are some different ways that we can produce more copies or make more copies of that DNA. So we can use DNA polymerase enzymes, and they make DNA. DNA polymerase enzymes are what our body uses to create more copies of DNA normally. Okay, so we can extract and use those enzymes if we're outside of the body too to make more copies. We can use gene synthesizers, which are an artificial way that we can make more copies of DNA. Or we can use the enzyme called reverse transcriptase to do reverse transcription in order to go from an mRNA copy back to a DNA copy in order to make more DNA for a particular gene of interest. So first of all, we'll look at DNA polymerase. So the DNA polymerase is an enzyme, which means that it's a protein, and it's required to synthesize nucleic acids. When we were talking about the trip operon, we learnt about RNA polymerase and how that makes small copies of the structural genes in that operon. DNA polymerase, on the other hand, it makes DNA. So it's the enzyme for DNA synthesis. So in order for that DNA polymerase to do its work, it needs the DNA, first of all, to be unzipped and single-stranded. Okay, the whole thing doesn't have to be single-stranded. We just need a single strand, a template to be able to use. Then the DNA polymerase will go along that single strand and make a complement. So we can see in this diagram just here, DNA polymerase doing its work here. It's going along the DNA and making a new copy. It's important for us to note that while we need to know that DNA polymerase is used to make more copies of DNA, when this happens naturally in our body, there's lots of other enzymes involved. Fortunately for us with our study design, we only need to know that DNA polymerase is responsible for making more copies of DNA. But just for your reference and to help this to make a little bit more sense, we've got a couple of other enzymes that are required. DNA helicase unzips the DNA. Primase is important because it signals where the DNA polymerase can begin the replication process. They're two important enzymes that happen in that process naturally, just to be aware of. Our second technique that we use to make DNA is through a gene synthesizer. So the gene synthesizers are basically a really big machine that string nucleotides together based on some information in the computer that tells them what sequence to put. So they're an artificial technique that we can use to quickly make new genes. A limitation is we can only do this with small genes, not with very large ones. And finally, we can do reverse transcription using the enzyme reverse transcriptase to go from an mRNA strand back to a DNA strand. So it's reverse transcriptase because it's doing the reverse of transcription. We go from mRNA back to DNA. This is just a diagram showing what's happening with reverse transcription. So as I said before, we start with our mRNA and we end up back with a DNA strand. Here's a question for you to answer on your Edpuzzle. We're now going to move on to restriction enzymes and look at how they cut DNA into fragments. Restriction enzymes can also be known as endonucleases. Okay, they both mean the same thing. They are enzymes that cut. So these enzymes cut at specific recognition sites. So they will only cut at a specific sequence. There are many different types of restriction enzymes, and I'll show you some examples in a moment. But essentially what you need to know here is that these guys, their job is to cut DNA at a specific site. Most restriction enzymes have a recognition site that is 4 to 8 base pairs long. So an enzyme with 4 base pair recognition site will cut the DNA around about every 256 base pairs, but 6 base pair recognition sites only cut at around every 4,096 base pairs. So these 4 base pair recognition sites are more common because they cut more frequently, which is often what we're looking to achieve when we're manipulating DNA. So just for your reference too, there's many different restriction enzymes and their name is based on where they've been derived from. So for example, one that we're going to see lots in examples and questions is ECO-R1. Okay, so ECO comes from the E. coli bacteria. So they just take the E and the first 2 letters of the second word. Okay, so just for your reference, these restriction enzymes are often extracted from bacteria. These are some examples of restriction enzymes, the organisms that they've been derived from, and what their recognition sites are. Luckily, you don't need to memorize all of these, but we will look at their action in more detail shortly. There are 2 types of restriction enzymes. There are sticky and blunt end cutters. What that means is some restriction enzymes will cut DNA to produce what we call sticky ends. And sticky ends occur when we've got overhang of DNA. So you can see here that this enzyme, HIND3, has cut this particular piece of DNA so that we've got some TCGA exposed on one side and then the overhang is on the other side. Okay, if we go down the bottom here, Echo RV, it is a blunt end cutter. It simply cuts the DNA straight through the backbone. So you end up with two pieces of DNA with no overhang. There are some advantages and disadvantages of both sticky and blunt end cutters. Sticky end cutters are usually more advantageous when you're trying to anneal or easily stick together strands of DNA because they'll easily stick with their complement. A disadvantage is if you want to stick DNA from two different sources where you need two different restriction enzymes, that's not possible in this case. You can only join two pieces of DNA together if they've been cut with the same restriction enzyme. So that's a limitation to this process. Okay, we can see two different pieces of DNA, a pink one and a green one. They have both been cut with the same restriction enzyme and therefore they've been able to easily join and anneal together. The word join and anneal are the same. Anneal just sounds better in our biological context. With the blunt end cutters, the ones that cut the DNA strand right in the middle, okay, some advantages of that is if you're trying to join DNA from different sources, okay, and you don't have matching restriction enzyme sites, you can use these, okay. Disadvantage is it's much more difficult to stick these or religate these back together because you haven't got any of that overhang. And also what can happen with these guys is that you can end up having a piece of DNA that's going this way and a piece of DNA that's going this way. If they're cut at a blunt end, you might find that they end up orientating the wrong way. So the result you're trying to achieve with joining them could go wrong for you. Okay, with these restriction enzymes, we're just going to delve a little bit deeper and just start to think about visually what happens when we cut DNA. So if we've got a piece of DNA that is 3,000 base pairs long, and we cut that DNA once at 500 base pairs and another time at 1,500 base pairs, when we cut, we're going to end up with a piece of DNA that's 500, that's come from just here, a piece of DNA that is 1,000 base pairs long, that's come from the middle there, and a piece of DNA that is 1,500 base pairs long. Okay, so our linear piece of DNA that we've started with, that we've cut twice, ends up forming 3 fragments of DNA. Here's an example question that you can have a go at on Edpuzzle. Our final type of tool for this video is a ligase. So ligase are enzymes which join DNA fragments back together. So ligase is DNA glue. So we can see here that we've got some fragments of DNA which have been cut with restriction enzymes to produce 2 fragments. We've then used our same restriction enzyme to go off and cut another piece of DNA. DNA ligase is simply used to glue those 2 pieces of DNA back together by joining those hydrogen bonds between the complementary base pairs together and joining together the phosphate sugar backbone. Okay, this is just another diagram to show you we've got our red DNA and our green DNA. The ligase is used to join that back together. And here is your final question for your Edpuzzle video today. Thanks everyone.